And it's really a pleasure to come and speak with you all today. So by matter of prefix, I'm a lung cancer oncologist, and I've actually had the benefit of actually working with some of the principles that Wendy has highlighted in the context of a specific disease and with specific patients. So what I'm going to do today is speak about my experience in oncology and in this specific disease, about how we're integrating genomics into the care of patients and some of the visions for the future, because we really have made dramatic strides with this model system. I'm also going to primarily kind of speak about this concept that we now have novel technologies that can look for some of the same genomics, meaning rearrangements, somatic mutations, amplification events in some cases, in plasma. And this creates a unique opportunity because it really opens up the door to how we can integrate this knowledge into the treatment of patients. So here are my disclosures. So as a matter of preface, I think it's very important that we understand this disease as we understand actually how we integrate the novel technologies in the treatment of patients. As a matter of preface, it was in 1994 that chemotherapy showed benefit in lung cancer over best supportive care. That's not so long ago. So 1994, that chemotherapy comes on the stage for lung cancer. It was, it was in about 2000 that we knew that giving a second line of chemotherapy after an initial line of chemotherapy showed benefit. So that established in lung cancer at least this concept of sequential therapy. It was in 2002 that we assessed that different chemotherapeutic agents can be given in the front line that set the stage for what is traditionally kind of known as the standard of care in lung cancer, which is a doublet, platinum-based doublet chemotherapy. And it was in 2004, which on the heels of prior studies over the earlier in the early 2000s, that the drug erlotinib, which was known as an EGFR inhibitor given to all lung cancer patients, showed really great efficacy for a select patient population. But no one really knew who. And in fact, actually, people hypothesized that the patients who would get the most benefit would be those patients that expressed EGFR by IHC in their tumors. And in fact, actually, when that biomarker was selected, in fact, actually, no efficacy over placebo was seen. But they hypothesized then actually that maybe there's mutations or something that is conferring the sensitivity. So actually, there were three groups, almost synchronously across three different institutions, so Mass Gen, Dana-Farber, and Memorial Sloan Kettering, that published that the patients actually who were getting the best benefit had activating mutations in EGFR. And the canonical mutations that are represented are an exon 19 deletion or an L858R somatic mutation. So that was in 2004, 2005, about 10 years ago. So in fact, actually, for the last 10 years, the standard of care for a patient who has that specific mutation is a pill-based therapy. And those patients now are living for actually many years. In fact, actually, I had a patient who lived for 10 years with lung cancer, metastatic stage, with this specific mutation. On the heels of that, there have been other genomic events identified, and we're going to kind of speak a little bit about those as well. So lung cancer historically has been a disease that has been associated with smoking. We know that, in fact, actually the most common lung cancer at one time was small cell lung cancer, but because of changes in smoking patterns, now non-small cell is more common. I have two pie charts kind of represented over here. The one on the left actually kind of shows patients actually in whom have smoking histories, and the pie chart on the right actually patients who don't have smoking histories. And what we see is that the genomic constitution of a patient who develops lung cancer in the context of smoking is very different than a patient who is a never smoker. In fact, actually, the most common mutation in a smoking patient population is the KRAS mutation. Next most common is EGFR, and then there are a host of different fusion events, for which there has been actually active drug development and even FDA approvals with pill-based therapy. So now if we look at patients who have had never smoking histories, we see that the KRAS population is actually quite a bit smaller. In the EGFR patient, it takes up about 50%. In fact, actually, there's some ethnic distributions as well. Patients actually in East Asia who develop lung cancer have about a 50% chance of having an EGFR mutant lung cancer irrespective of smoking. We also know that EGFR mutations are more common in women, patients of younger age, and in fact, actually had a patient who was diagnosed with lung cancer during pregnancy at age 33, also with the EGFR mutation. So on the heels of this, what we also know is that patients who get the drugs that are specific for that mutation in the context of EGFR are doing better. What we see is that on the upper right box, if a patient is EGFR mutation positive and gets the drug gefitinib, which is an EGFR inhibitor, they do better than chemo. But also even more striking, if a patient is EGFR negative and gets the drug gefitinib, chemotherapy dramatically outperforms. So one of the big lessons is giving a molecularly targeted drug to a patient who doesn't have that specific mutation is actually the wrong therapy and underperforms compared to chemo. So we really need to be precision-oriented about who we're giving these drugs to, not just using precision drugs. And that kind of establishes the fact, actually, that we need assays and technologies to select these patients. So we now actually, after 2005 and these drugs entered into the frontline use, we now have a better knowledge about why patients progress. And inevitably, there are few patients actually who are cured with these even site-selected therapies. So when patients progress, they usually progress actually for one of a few different mechanisms. So one could be additional mutations at the level of the gene, the same gene, EGFR. So new mutations in EGFR that confer differential drug binding. This is the same story actually that exists in the context of CML with second site mutations. There can be additional bypass pathways. There can actually be even histologic switch of the cancer from an adenocarcinoma to a small cell. And we think that on basis of selective pressure, there can be different programmed events through epigenetics and other types of strategies to confer that reprogramming event. Interestingly, we have also seen novel fusion events in these post-progression biopsies, which are important. So I want to highlight that all of this data, which came about in the early, 2010, 2012 time, was from repeat tissue biopsies. But we know in lung cancer especially, getting additional biopsies is very difficult. One has to, so firstly, on top of these therapies, there's cytoreduction. One's dealing with a low amount of tumor burden. Getting the biopsies at the time of small residual disease is actually very difficult. So how are we going to do this? And it really creates a challenge because we need to understand this. So here actually just shows a schema of how the resistance is actually happening. We see that resistance, again, is happening at the EGFR receptor. Downstream of the receptor, EGFR signals through the MAP kinase pathway and the PI3 kinase pathway. And there can be second site mutations both in the MAP kinase pathway as well as the PI3 kinase pathway, or bypass pathways that are upregulated. We also know that there is different biologies. I had a patient actually who progressed on the drug Tarceva or erlotinib within a few months of therapy. It's speculated that that patient had a resistance mutation from the beginning that grew out what we know is an intrinsic resistance or de novo resistance. Or actually, there's also been the context of patients, and I've had a patient actually who for 7 years had no progression and then had progression. So there's this hypothesis that there is a drug tolerant state that's primarily driven more by stem cell pathways, and Jeff Engelman at MGH has kind of demonstrated this preclinically, that on top of drug, there can be this persister that then actually acquires this selective advantage through mutation that can then launch out at a different time. So understanding this and understanding the different biologies about how this is happening really kind of forces us to understand how are we going to track patients and how are we going to be able to survey people over time when the mutagenesis can happen in such differential ways. So here are the drugs that are approved now. Since 2005, when the drugs gefitinib and erlotinib were first in the picture, again, those are first-generation EGFR inhibitors. We now have second-generation inhibitors that were initially created to target the resistance with T790M, but in fact, actually, pharmacologically didn't achieve that. We now have an FDA approval for a third-generation inhibitor, which targets the original activating mutations, the exon 19 deletion and L858R, plus the resistance clone, the T790M, which occurs in 50% of the patients. So this is now FDA approved, and there are studies actually looking at this now in the front line. So not giving it post-resistance, but giving it pre to try to ward off resistance from happening. So this is kind of my final slide as a context of lung cancer, because it's my belief that if we understand this context, understanding how to integrate the technologies in this way will become a little bit easier. And this is framed around two patient cases. The first patient actually in 2007 was diagnosed with lung adenocarcinoma, had the L858R EGFR mutation, and got chemotherapy. With chemotherapy, because chemotherapy is still an effective drug, there was some reduction, and at that time, the drug erlotinib was being given as a maintenance type of therapy to try to maintain the cancer down post-cytoreduction. And there was stability of disease with that for about 1.5 years. But then that patient progressed, developed resistance, and had a repeat tissue biopsy. And at that time of biopsy, we see that the patient has a new mutation in EGFR, the T790M, which exists in about 60% of cases, and the patient progressed. At that time, because we didn't have any other novel agents or targeted resistance type therapies, the patient got chemotherapy again, had some amount of stability, and what we see is that off of the drug erlotinib, the clonal dynamics in the tumor have changed based on a repeat biopsy post the chemo. Now the resistance has gone away because the drug has been stopped. And so off of the selective pressure of erlotinib, the clonal dynamics change where the T790M is no longer detected. That patient was then retreated with erlotinib, and actually we see that the cancer kind of reduced. So the second patient below, also a patient with adenocarcinoma, the L858R mutation, got erlotinib as a front line. We see that there's reduction in the cancer. The patient progresses. At progression, histologically, the cancer has switched now to a small cell lung cancer and has the PI3 kinase mutation as a resistance mutation as well, in addition to the original EGFR mutation. The patient then gets chemo and radiation. So off of the erlotinib, the clonal dynamics within the tumor change, where that resistance phenotype, again, the small cell PI3 kinase and EGFR-driven cancer, changes in a way in which the original EGFR clone now populates more than the resistance. And we see based on another biopsy that the cancer is now adenocarcinoma with the L858R mutation. The patient then gets erlotinib, and there's reduction, albeit for a shorter period of time, and then the patient progresses and has that same resistance phenotype with the small cell and PI3 kinase and L858R. So what this demonstrated to me is the fact that it's very important that we are tracking patients through time because the biology is changing on top of the drugs. And if we're able to track patients, then we can preemptively understand how that biology has changed and kind of adjust the therapy accordingly. So how are we doing this in real life now in practice? So, as Wendy had mentioned, actually, kind of genomics has revolutionized this disease. It's identified novel targets. It's also kind of led to active drug development. Much of our knowledge of how we treat patients is based on tissue rebiopsy efforts. Tissue rebiopsy actually confers a significant amount of cost, operator dependency. There can be complications, much, in the case of lung cancer, include pneumothorax, infection, such. And so a recognition, actually, that if there was a non-invasive or less invasive way to survey patients through time for these genomic events, actually is very important. So our knowledge of doing this non-invasively, actually, at least from a genomic standpoint, has come a lot from prenatal testing. Actually, in the prenatal world, looking at circulating fetal DNA in a pregnant mother actually has been very helpful in understanding the distribution of differential fragments of DNA in plasma and how that can be used to track kind of origin as well as genomic events. So what is cell-free DNA? So all of us in our bodies, we have circulating fragments of DNA that circulates in the plasma. Those fragments actually are typically in the order of about 180 base pairs. That's largely because the DNA gets degraded through time and on basis of nucleases. And when we're exercising, we have a lot of cell-free DNA circulating. If we have an infection going on, we have cell-free DNA. In a patient who has cancer with a certain amount of catabolism and increased energy needs and such, that patient also has circulating tumor DNA that's circulating around. That circulating tumor DNA can be tracked by virtue of looking at the sizes of the fragments of DNA that exist. Typically, the DNA is about 180 base pairs. And when one can find specific genomic aberrations specific for the cancer on that cell-free DNA, we call that circulating tumor DNA because it's specific for the fragments of the DNA that's coming from the cancer. And now one can think about what are the implications of this? Can this be used actually for diagnosis? Can it be used for monitoring? Can it be applied as a biomarker? And I'm going to speak about examples about how this technology has been used in that way in the context of lung cancer. So the four primary ways in which circulating tumor DNA is being evaluated, one is early detection. If we have presumed normal, healthy patients, people like you and I, if we have a blood test and if we can diagnose that there is a genomic event there, can that give us an idea about do we have cancer or an occult cancer, maybe kind of an asymptomatic manifestation of malignancy? And again, that would be the holy grail, because if we could find that, then actually we know that the only way to cure a cancer is actually if it's surgically resected or kind of removed, eradicated through radiation or surgery. The next major kind of application of circulating tumor DNA is around rapid genotype testing. So this is actually in patients in whom have a context for needing to be evaluated with a genotype such as lung cancer with EGFR mutations, ALK rearrangements, and such. So if we can do that rapidly, that could be very helpful because doing a tissue biopsy really requires accessioning the tissues out of pathology, sending over, getting it sequenced. That whole time can take about 2 to 3 weeks, even in some cases more. Presumably by a blood test being sent over and analyzed, that could actually speed the time to a result. Resistance genotyping. So we spent a lot of time talking about resistance and kind of the fact actually that cancers are changing through time. If we're able to survey patients with a blood test over time, we could generate that data. In those 2 patient cases that I described, actually we could do that not with repeat tissue biopsies, but with a plasma test. And then some early detection of response. And we'll speak about this too, because what we've seen is that by changing the amount of cycling that's happening within cells, that influences the amount of DNA that's shed. So one can get an early surrogate of actually response by virtue of tracking the cycling based on the drug and the amount of DNA that's released. It's important to note that we've been talking a lot about plasma, and I'm going to present mostly on plasma, but these fragments of DNA from cancer can be found in other fluids. They can be found in ascites or pleural effusions. They can also be found in urine, and there's ongoing efforts to really characterize those substrates because again, we can always envision that, a substrate like urine has a, very non-invasive way to collect. And it also influences the frequency of collection and the volume of collection. Because if we're able to increase our volume, then actually we have a better chance of detecting rarer fragments. It's also important to note that the fragment sizes as it exists in each of these locations of plasma, urine, ascites, and such is also very important because the technologies need to be specific for actually being able to, to characterize long and small fragments. I think Wendy alluded to this as well, is that specific technologies are built in different ways, and those are important to characterize about what the applications could be. So the major technologies that are being employed for circulating tumor DNA are digital PCR or next-gen sequencing. So I kind of just put some bullet points about major kind of characterizations of differences across these two. Again, actually, digital PCR it's best for finding somatic mutations. It can be used for copy number analysis as well. We need to know what mutations we're looking for, though, when we use digital PCR, because the primers and the technologies are built around those specific that knowledge. The sensitivity is dependent according to each gene that we're looking at, and that's important because one may get a result from an assay that says, well, we have this sensitivity, but that may not be uniform across each of the genomic targets that we're looking for. That may lead to some element of bias about what we find. It's generally fast and reproducible, minimal bioinformatic needs, and that's important when we think about cost. So now actually next-gen sequencing, again, actually this is important because we don't necessarily need to know what we're looking for. It allows the capacity to kind of for discovery, and for a patient actually who we may not know what the genomic events are involved with, this is important. Actually, it can facilitate rearrangements, amplification events, whole exome, whole genome sequencing can be done. It does require more stringent bioinformatic needs for error suppression, and that actually can increase expense and time. So here is a schema which shows one of the technologies actually that was pioneered at Johns Hopkins called TechSeq. And again, this is actually sequencing at 30,000x, so very deep sequencing, because again, we're looking for very few fragments in a tube of blood out of about 10,000 genomic equivalents. So the way that these technologies work generally is that there's cell-free DNA that exists in the tube of blood. That cell-free DNA is barcoded. That barcoding actually creates a certain amount of labeling for each of these fragments. There's redundant sequencing, and on basis of the hits actually that are consistent across multiple redundant sequencing events versus more isolated events. And again, that's depicted actually in the fourth kind of component of the schema. That's a way where one can weed out what is signal to noise, like what is actually truly a true mutation versus an artifact of the sequencing events and the redundant sequencing. And then actually there is reference to there's alignment to the reference genome, and then actually the reporting of results. So, these types of assays actually are being created. They can be as comprehensive as like 64 genes. In some cases, actually, through research use, people have been able to do whole genome sequencing, actually in plasma as well. Or they can be more limited, and that has different costs and implications. So here actually just shows kind of the bioinformatic difference between with the barcoding and the deep sequencing. One can really kind of get down to 0.01% error detection rate. Okay, so this was one study actually that we were involved with in which we were using this specific assay and trying to see, can we actually find cancer at its earlier stages, stage 1 and 2? So the way that this study was actually arranged is patients actually who had stage 1 or 2 malignancy, and the malignancies that were looked at were lung, ovarian, and breast, and colon. There was one cohort of early stage patients, one cohort of more advanced stage patients, and then one cohort of normal, healthy people who do not have cancer. And we looked using the assay to see what was the differences in our ability to detect cell-free DNA across each of these cohorts. So we can see that for healthy patients, as well as patients with cancer, we can detect cell-free DNA. And that's a true known fact. We all have cell-free DNA circulating. But now actually when we look for specific mutations according to that panel that I kind of showed before, when we look at normal healthy patients, we do not see any of the genes speculated in cancer in those patients. In stage 1 and 2 patients, we were able to detect circulating tumor DNA, as well as in late stage patients we were able to detect. So by virtue of 30,000x deep sequencing, as well as the error correction, one can actually find circulating tumor DNA in patients with early stage. So again, this is a good proof of concept to show that we can find circulating tumor DNA in early stage patients. Now, if we look at this in large cohorts, can we use this to screen patients for early stage malignancy so that then actually at its earlier points, patients can have resections and such. And that's where this work is going. So here actually just shows that based on that large analysis of over 200 patients, the frequency of genes across lung cancer, breast cancer, and colon cancer were synchronous with what's seen in the COSMIC database. And then actually, this was actually compared to tissue biopsies. And in some cases, actually, the specific genomic events were found in plasma and not tissue, and vice versa. It's actually important to note that actually in patients who were found in plasma, there was ultra-deep sequencing done in tissue across multiple regions of the tissue as well. And in several cases, actually, they found the variant that was then in the plasma. So it really kind of leads to this concept that plasma gives us a global understanding of biology across metastatic sites and even intratumoral heterogeneity. Because as we know that tumors are, across the spatial conformation of a tumor, there can be different genomic events in different components of the tumor. And based on a biopsy, if we're just biopsying one specific area, we may not have that more comprehensive understanding. Okay, so here is some work actually about looking at circulating tumor DNA in patients actually who got treated with therapy. So what we see is that when patients are treated with erlotinib, there can be reduction in the EGFR clones. In fact, actually, people had worse prognosis if you were still able to detect the EGFR circulating tumor DNA after therapy. So again, finding it after therapy means that there's some residual disease and patients are doing worse. Here shows actually how, why this is relevant. We now have a drug actually that works really well. In fact, actually, this drug, osimertinib, every column below the 0 point actually shows a response, and actually below 30% is what we call a partial response. And so across dose, this drug is working very well. And we now know that through sophisticated genomic type studies, we can actually assess whether or not concomitant genomic events are existing on the same fragment or on different fragments, and that has implications to kind of understand the heterogeneity of the tumor and the different clonal events there. And so this actually just shows how each clone can be tracked vis-a-vis others through time based on drug. So here actually shows, the rise of a second site mutation in EGFR, as well as different other clonal events actually which can be detected outside of EGFR with PI3 kinase mutations, and how that links into the radiology. We see that those patients actually who retain bypass pathways are doing worse. And again, all of this data was accumulated through surveillance through plasma. So the ability to actually integrate this now into patient care is where we're at. And here actually, this was published in Journal of Clinical Oncology of an algorithm based on FDA approvals of how the liquid biopsy is being integrated into lung cancer. So if a patient has acquired resistance to an EGFR inhibitor, the traditional kind of way has been a tissue biopsy to assess T790M and then getting that third generation inhibitor. We now actually have an FDA approval for a plasma test where based on the plasma T790M detection, if a patient has a plasma positivity, that patient can go on to get the third generation compound. We still recommend, and I still recommend getting the tissue biopsy because there can be a lot of clonal heterogeneity. And also there is still some genomic events that can be detected in tissue, better or kind of on panels, more reflective than plasma. And also kind of this concept that the cancer can switch histologies. So for that reason, integrating both a tissue as well as plasma may be an answer rather than one superseding another. Okay, so I just want to kind of end actually highlighting the fact actually that there have been efforts to kind of expand out beyond plasma, looking at urine. This was a study actually that compared tissue, plasma, and urine together for EGFR mutations. And what we see is that in a total of about 181 cases, there was 104 cases that were synchronous in which all three detected the same genomic events. So, that can be seen with both, kind of in an optimistic way as well as a negative way. I mean, it's not capturing the full consistency, and we're going to have to develop, strategies to incorporate different ways of, getting a more comprehensive data set. So here's some work actually that we showed actually in a pleural effusion, being able to detect amplified EGFR, and this was actually very important, for patients actually who, have that because we can also get the cells, and those can then actually be used for patient-derived xenografts and such. And this was a patient in ascites actually that we were able to detect a PI3 kinase mutation as well as amplification events, and actually chromosomal rearrangements, and this is more reflective of what's called chromothripsis, or shattering of the DNA. So in conclusion, actually, circulating tumor DNAs have been used for proof of concept for early detection of cancer. We need larger studies to validate this across larger populations. The initial genotyping in lung cancer is prime time. It's currently being used by lung cancer oncologists or commercial assays, which are doing this as a service. Monitoring patients, so meaning the repetitive evaluation of circulating tumor DNA, is also being done, but again, and can give us some knowledge of resistance mutation as well as tracking. But again, larger cohorts are needed, and this becomes of value if we're able to align differential therapy decisions based on the earlier detection of resistance. So the types of clinical trials that are being brought forward are integrating combinations early based on the detection of the resistance. We spoke a little bit about how different substrates are also being used in composite to give us a more comprehensive view, and that work is ongoing. And we still have to see kind of what the true fruition of utilizing that more comprehensive approach will be. And then actually it's important that while I spoke about this in lung cancer, and for a long time lung cancer has been a model of integrating genomics, such strategies are being applied to colon cancer and breast cancer, and other cancers as well. So kind of it's gaining momentum across oncology. So with that, actually just want to thank you all for listening, and thank actually people that I work with actually to facilitate and work on these projects. So thank you.